Tissue-resident memory T cells can sense the physical feel of the tissue they occupy, not just the chemical signals around them. That McGill-led result means long-term immunity is not only a chemical conversation. The mechanics of the organ itself can shape the cells that stay behind to protect it. This is Planetterrian Daily, episode 155. These are the T cells that remain in tissues after an infection has cleared and respond rapidly if the same threat returns. For years, the field treated that population as a product of cytokines, antigen, and local chemical cues alone. The McGill University-led work, reported by phys.org, adds a physical dimension. The cells appear able to sense touch, meaning the mechanical character of the tissue can shape them. The finding stops at the existence of that tactile capacity. The receptor, the mechanical cue, and the transduction path are still unnamed. If resident memory can be shaped by how a tissue feels, vaccine and immunotherapy design may have a second control besides the chemical adjuvant. Antigen and the cytokine milieu still matter. So, on this evidence, do the stiffness, stretch, and texture of the organ the cell is asked to occupy? Tissues that remodel with age, a denser matrix, different mechanics, may not build the same resident memory even when the antigen is identical. That would make part of immune aging a mechanical problem rather than only a molecular one. The study cannot yet say which tissues are most affected or how to measure the signal in a human biopsy. The next result that matters is how touch is transduced and whether that pathway can be read or nudged in human tissue. Another kind of molecular memory showed up in a separate report, not in the immune system this time, but passed from mother to child. In humans and many other animals, a mother's age can affect the physical and behavioral characteristics of the next generation. Scientists call these changes maternal age effects, and the pattern is widespread across the animal kingdom. Researchers still do not fully understand how or why it occurs. A new report, also covered by phys.org, treats the effect as molecular memory, traces of the mother's age that show up in the offspring cells, not only in the mother. We covered aging biology yesterday. This does not hand us a human biomarker. Naming the molecules that carry that echo and testing whether they can be read in human tissue is the measurement step still missing. From a mark the body keeps without asking, we turn to a mark people carry for years, and a method that tries to loosen it. Childhood criticism can leave a fear of failure running well into adulthood. New work covered by Science Daily found those emotional patterns may be more flexible than they feel. Young adults who revisited painful memories through imagery-based therapy showed lasting reductions in fear, guilt, sadness, and stress. The method does not ask people to suppress the memory. It asks them to go back into it, in imagery, and let the feeling attached to it change. The sample is young adults, and the target is fear of failure, not a formal anxiety diagnosis and not an older cohort. Durability beyond the study window and a test in people who meet clinical criteria are still open. You've probably heard that memories are stored like files in a cabinet. Write once, close the drawer, pull the same folder out later, unchanged. That picture is wrong. The brain does not file a finished object. It stores a trace that has to be rebuilt every time you use it, and the act of rebuilding briefly makes the trace unstable. Neuroscientists call the first locking down consolidation. They call the later, recall-triggered reopening reconsolidation. Right now, as you picture the room you are sitting in, or the last conversation that stung, the networks holding that episode are temporarily writable. New proteins have to be made to lock the trace back down. If that protein-making step is blocked in the laboratory, the classic demonstration used a protein synthesis inhibitor in the amygdala of rats after a single reminder of a learned shock. A fear memory that had already been consolidated can fail to return. The open window is often put at about 4 to 6 hours. That is long enough for a conversation, a therapy session, or a night of rumination to rewrite the feeling attached to the event, and short enough that by the next day the updated version is what remains. The conditions for opening the trace are stricter than a casual recollection. A strong old memory does not always unlock just because you thought of it. Many experiments suggest the brain needs a mismatch, a prediction error, before it treats the trace as worth updating. That is one reason a vivid, structured retelling can change a fear, while idly turning the same scene over in your head can cut the groove deeper. Unstructured replay often restates the old prediction, so the window never really opens, or it opens and then restabilizes the same emotion. Imagery-based work, including the fear of failure study just mentioned, tries to use that window on purpose. The person is asked to re-enter the scene in enough sensory detail that the memory comes online, then to let a different ending, a different body state, or a different meaning attach before the proteins lock it again. None of this means every painful memory should be poked. Boundary conditions matter. Very recent trauma, dissociation, and some medications that interfere with protein synthesis or attention can change whether reconsolidation is even in play. If you are going to recall something difficult, how and with whom you do it is part of the biology, not a side issue. A structured session with someone who knows the method is a different intervention from replaying the insult at 1 a.m. Watch for trials that test whether reconsolidation-based methods still hold months later in older adults and in diagnosed anxiety, not only at the end of a short protocol in young volunteers. That durability test is how you tell a rewritten trace from a good week. From how a living brain rewrites a feeling to how an extinct one forces a rewrite of something we thought we knew about bodies. A new study by the Department of Anatomy and Anthropology at Tel Aviv University, published in Scientific Reports, takes up one of the striking differences between men and women, how the modern human pelvis evolved. Male Neanderthal pelvises resemble those of modern females. That comparison challenges decades-old assumptions about which pelvic shapes count as male or female in our evolutionary past. The argument is built from fossil anatomy set against living human pelvic form, not from a genetic reconstruction. Additional fossil measurements will show whether the resemblance holds beyond the specimens used in this paper. A finding about deep time, then a finding about right now. A plant product and a poison center curve that got away from the guidance. Kratom use and serious health problems linked to it have risen dramatically across the United States over the past decade. Poison center reports jumped from 19 cases in 2010 to more than 1,200 in 2023. Severe outcomes, including life-threatening complications, disability, and death, also climbed sharply. Researchers warn that kratom can trigger seizures, abnormal heart rhythms, liver damage, and breathing problems, with risks potentially increasing when it is combined with other drugs. The case counts come from poison center reports, which capture people who were ill enough for someone to call, not the full user population. Labeling, scheduling, or clinical guidance catching up with that volume is the public health question the numbers put on the table. From a product already in wide use to 2 delivery ideas still on the bench, vehicles that might someday carry a therapy. Red blood cells serve as the foundation for nanocarriers that a new study tested as vehicles for gene therapy, tumor targeting, and other medical treatments. Scientists at The Ohio State University engineered extracellular vesicles on that template and showed that the vesicles could evade immune cells and target cancer cells. Those are 2 jobs that delivery vehicles usually fail at. The study is published in Advanced Healthcare Materials. What the paper establishes is a vehicle, not a finished medicine. No therapeutic cargo is described as having completed an animal efficacy test here. Loading a real payload and taking that construct into animals is the experiment that would connect the design to in vivo delivery. Another remote control idea for genes, this one from outside of the body, with a sensor instead of a cell-derived shell. The DNA of living organisms contains regulatory elements that control when, where, and to what extent specific genes are turned on or off. Those elements can be co-opted to create gene switches, tools for understanding gene expression and for therapeutic applications, particularly the non-invasive treatment or management of genetic disorders. A new report, covered by phys.org, describes a molecular electromagnetic sensor that may enable remote-controlled gene therapy, using those switches from outside the body. External electromagnetic control is the proposed route, not a chemical inducer the patient has to take. Demonstration in living tissue with a safety profile attached is what would move the sensor from a laboratory idea toward a therapy. Delivery and control are the hopeful half of gene therapy. The other half is what happens when high-dose viral vectors go wrong and no one says so. 2 recent patient deaths have drawn international scrutiny to China's investigator-initiated trial framework, the IIT path used as an alternative regulatory route. Both deaths involved severe immune reactions to high-dose viral vector-delivered gene therapies, and both came with a lack of disclosure. The Fierce Biotech report describes what appear to be systemic gaps in oversight and transparency on that path. High-dose viral vectors are already known to be capable of triggering dangerous immune responses. The added issue here is that the deaths were not disclosed in a way that satisfied outside observers. Whether IIT disclosure rules change is the concrete follow-up. Next time, we'll be watching for whether those disclosure rules actually move, and for the paper that names how a resident T cell transduces touch. That covers today's science and health news. Share this with someone who's curious about the latest research. I'm Patrick in Vancouver. See you next time. And if you'd rather watch than listen, find us on YouTube at NARA Network. Links in the show notes. One more thing. If you liked today's episode, our sister show OmniView is worth a spot in your feed. A balanced daily world news briefing that takes every side seriously. It's one of the NARA Network's daily briefings, all free at naranetwork.com. Live dashboards, TSLA, SpaceX launches, the investing scoreboard are all at naranetwork.com/data. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.